Within the sheep industry, there are many issues that can cause a huge impact on production. One of these issues is the gastrointestinal nematode parasites that affect the health of the animal. It has been discovered through microarray research that sheep contain genes that allow the animal to be resistant to such parasites through the immune system. However, could the animals also be avoiding the pasture that has a high risk of containing the eggs for these parasites? As sheep are in a pastoral environment, localized concentrations of forage resources and parasites are together due to the deposition of feces. The animals generally avoid the feces, creating a higher concentration. This concentration is predominantly tussocks of tall grass, which cause the animal to risk parasitism by consuming the nutrient-rich tussocks of grass. The report, Genetically Resistant Sheep avoid parasites to a greater extent than do susceptible sheep, studies this variable. This study was created to see if the population of nematode parasites within the environment were avoided by more resistant sheep than the susceptible sheep. The animals within the experiment were selected through 13 years of research. This research used the Parindale breed and selectively bred the animals that proved to either contain or not contain the genes for immunity through microarray research that had a high or low fecal egg count. The experiment was conducted over a 12-week period, with the feces sampled weekly and the animals' behavior monitored to obtain results. The tussock and intertussock within the 1 hectare paddock were grown in a mosaic pattern found in natural systems. The behavior was then monitored for 3 weeks, the number which being the number of bites, the bite rate, and the number of steps taken by the animal. The length of the tussock and the intertussock were observed. Through observation of behavior, it is seen that the R sheep made almost no attempt to graze at the tussock, unlike the S sheep. Figure 2 shows this through the amount of bites taken of the tussock. At the end of the experiment, the FEC showed that the R sheep had less fecal egg count, seen in Figure 1. However, because contact with the parasites is inevitable within any system, the better immune function of R sheep gives them the advantage over S sheep. This experiment may seem to be counterintuitive, as the S sheep may lack the nutrients required to fight off parasites due to the avoidance of tussocks, while the R sheep don't have that issue. To help understand the scientific theory behind genetic difference within sheep for resistance, reports such as microarray analysis of selection lines from outbred populations to identify genes involved with nematode parasitic resistance in sheep, and the use of genetically resistant sheep to control nematode parasitism can be viewed. These reports go in depth of how genes were located and how they affect the immune system of the sheep.